Hey, Chapter 12, Lecture 4 already. We are cruising right along through our understanding of the central dogma. And in the last lecture, we talked about DNA replication, and so that would be this blue arrow here. It's DNA producing DNA. So DNA producing DNA is replication. DNA replication happens in the nucleus. We've gone through all the enzyme steps. We've talked about helicase, unzipping and unwinding the DNA molecule by breaking those hydrogen bonds. Talked about primase laying the RNA primers, which gives DNA polymerase III a starting and ending spot. We talked about the single-stranded binding proteins that are going to hold that replication fork open to keep the DNA molecule from binding back up on itself or from creating those hydrogen bonds. And in this lecture specifically, we're going to talk about RNA transcription. So we're going from DNA that may or may not have been just replicated, but now we're going to go from DNA to RNA. Notice that DNA makes DNA in the nucleus, but DNA also makes RNA in the nucleus. Okay, DNA, if you're using something or some process that involves DNA, it's a good assumption to make that that DNA is going to stay in the nucleus all the time. You don't want DNA to travel out of the nucleus into the cytoplasm, because the cytoplasm is like the wild, wild west. Right? It's dangerous out there. There's enzymes everywhere. There's bacteria everywhere. There's viruses everywhere. There's things that the DNA doesn't want to encounter in the cytoplasm. And there are things that you don't want the DNA to encounter in the cytoplasm. So we need to take this really important DNA molecule, this really important item in DNA, and we want to replicate it so that eventually this cell can produce two cells. We've talked about mitosis, and we know what that is. Right? We have to copy the DNA before we make more copies of the DNA or more copies of the cell. But DNA is too important to send that molecule out into the cytoplasm. And so what do we need to do? We need to take this really important molecule and we need to make a copy of the section we want to express, meaning we want to make a copy of the gene or genes of your DNA that will eventually make a protein, so that we can ultimately use that recipe called RNA to make a protein in the cytoplasm. And ultimately, that's why you have DNA. Okay, what is DNA for? It's to make proteins. Let's talk a little bit about what transcription is and what this first step of protein synthesis is. Central dogma includes replication, transcription, and translation. But the protein synthesis portion of the central dogma is just transcription and translation, and so DNA doesn't really, or at least DNA replication doesn't really have a role in protein synthesis. But we've just established that DNA cannot leave the nucleus, okay, and we can't make proteins in the nucleus, and so we have to get the message from DNA, the message that's contained within our genes that are located in the nucleus of a cell. We need to get that material, or at least that message, from DNA form to RNA form so that we can eventually read it and produce a protein that will eventually be expressed. Okay? Those proteins are made and used in the cytoplasm, and they're made by a ribosome. Okay? Ribosome is this structure right here. A ribosome is made up of two subunits. You have the large subunit, which is obviously the larger component. You have the small subunit, which is obviously the smaller component. And the large subunit is used to bind to tRNA, and we'll talk about what specifically tRNA is and what it does and what it allows you to do. But the large subunit binds to the tRNA. And if you looked at this diagram, you know that there are 3 binding sites, and so you can probably guess that those are the binding sites that tRNA molecules will bind to. This is the large subunit, and in eukaryotic cells, this large subunit would be 50S, and the small subunit would be 30S. Eukaryotic ribosomes are called 80S, or referred to as 80S ribosomes. So you have a 50S large subunit, and you have a 30S small subunit. And so all the ribosomes in your cells would be 80S ribosomes, right? Now, these binding sites have names as well, if you guessed it by now. This is the E binding site, this is the P binding site, and this is the A binding site. EPA. The binding sites are really, really important when we talk about the growing protein chain. And we'll get into what actually happens within those binding sites later when we talk about translation. But for now, know the structure of the ribosome. The ribosome consists of two subunits, the small subunit, large subunit. The large subunit binds to the tRNA molecules. The small subunit binds to your mRNA molecule, and M stands for messenger, T stands for transfer. We'll talk about that in a little bit. But these binding sites play a real crucial role in ultimately the production of this protein. Okay, and so if this is our growing polypeptide chain, the growing polypeptide chain would be linked to the P site. E would be obviously like exit, and A is basically kind of like the activation site. We'll talk about that in a little bit. Our tRNA molecule is this. Okay, this is a tRNA. This top portion is an amino acid. The bottom portion is an anticodon. The anticodon is opposite of the codon. Okay, and so this would be our 3 nucleotide sequence down here. This would be the codon. The codon is located on the mRNA message, and the anticodon, like this picture shows, is located on the tRNA. But there are 3 types of RNA. You have mRNA, tRNA, rRNA. rRNA is the biggest component of the ribosome. Okay, it's called ribosomal RNA. tRNA, just like this molecule, is going to transfer amino acids from the cytoplasm to our ribosome in order to produce a protein. And mRNA, messenger RNA, is the actual recipe, so to speak, for our specific protein. Okay, so mRNA is produced in the nucleus. It is made, well, I should say all these RNAs are produced in the nucleus, but all of these RNAs are going to be produced by the DNA in a process called transcription, rather. And all of these RNA molecules are going to be single-stranded, and they have a different sugar than DNA. DNA has deoxyribose sugar. In RNA, it has ribose. So DNA stands for deoxyribonucleic acid. RNA stands for ribonucleic acid. And because RNA is a single-stranded molecule, we can't have this double helix structure because we don't have 2 strands to twist. We have a single ribbon-like strand that is linear, and the nucleotides that compose the RNA messages are going to be ribose instead of deoxyribose, and we're going to start using uracil, rather than T. So U is going to pair with 2 hydrogen bonds to A, and U is going to take the place of T, so that is uracil. If we talk about kind of what protein synthesis is, we know that DNA contains genes, and these genes are ultimately going to be expressed as proteins. But we can't just look at a gene and produce a protein. We have to take the genes in our DNA, produce a message called RNA, and then use that messenger, or use that message to produce the end result as protein. I like to use the analogy of a cookbook. Okay, imagine having a cookbook that was given to you by your great-great-great-great-great-great-grandfather or grandmother, and that cookbook is one of a kind. Maybe he or she produced it. They wrote down all the recipes. It is one of a kind. You cannot replace it if it is damaged. Well, it's similar to your DNA. You cannot replace your DNA. Whatever you have when you were born should be the DNA that you live with and die with. But we know that mutations can happen. Just like in that cookbook, pages can get ripped, pages can get folded, pages can get removed, pages can get smudged, burned, whatever. And so those are called mutations. It is safer for us, and using that cookbook, to open up the cookbook when it's in the safety of your kitchen or the safety of your house, copy down a recipe on another piece of paper that maybe is the best recipe for grilled pork chops. It's safer to take a separate sheet of paper, write down that recipe on that piece of paper, and then take that piece of paper to the store to get the materials you need, or take it to the grill and actually read it step by step as you're cooking the food. Okay? If that copy is damaged, it doesn't matter because you still have the cookbook copy. However, if you don't do the write it down step and you just take that cookbook to the store with you, or you just take that cookbook to the grill with you and it goes up in flames, you are in trubs because that is the only copy you have. and therefore can't replace it. So DNA is the same way. We want to keep DNA safe in the nucleus. We want to copy down DNA in terms of RNA, make RNA, which is basically disposable. Okay, we use RNA to produce a protein, and then we can throw it away or we can recycle it, but that's ultimately the goal. Genes we use to turn into RNA messages, which we then use to turn into proteins. Protein structure, remember we talked about primary, secondary, tertiary, and quaternary protein structure. That protein structure is dictated by the specific sequence of amino acids. An amino acid is the monomer, remember, of protein. And then based on how those amino acids are sequenced and where the disulfide bridges and the hydrogen bonds, where they're at, will dictate how that protein folds. Proteins will fold by putting polar amino acids together. They'll fold by putting nonpolar amino acids together. They'll fold by putting ionic and acidic amino acids together. They'll fold by putting hydrophobic amino acids together. They'll fold by putting hydrophilic amino acids together. Okay, amino acid sequence is the #1 dictator of how that protein will eventually look. Gene expression, which is how the gene is expressed physically, takes place in 2 steps. Like I said before, transcription is turning DNA into RNA, and then translation is turning that RNA into proteins. Transcription happens in the nucleus, translation happens in the cytoplasm. Okay. This is a picture of the process of protein synthesis. We take DNA in the nucleus. Here's the nucleus. Here's a nuclear pore. Okay. And we're going to take DNA, we're going to open up the DNA using RNA polymerase, which we'll get into. We will then take RNA polymerase, read the sequence of DNA nucleotides, produce an RNA message. We'll then do some RNA processing, which we'll talk about, where we'll cut out introns and we'll splice together exons. We'll put a cap and a tail on it, and then we will send it out of the nucleus through a nuclear pore, give it to a ribosome, and then let the ribosome use the mRNA, rRNA, and tRNA to eventually produce a protein through the process of translation, which will be folded based on the amino acid sequence and then ultimately what that protein will be used for. So let's talk about the nuts and bolts of translation. You'll notice that it appears to be similar to DNA replication, but we don't have all of the same kinds of RNA, or at least we don't have a lot of the same kinds of enzymes that we used in DNA replication. RNA polymerase is the main enzyme. Now that should be familiar to you because we've talked about DNA polymerase 3 and we've talked about DNA polymerase 1. Remember, DNA polymerase 3 is the enzyme that will produce the DNA polymer through the process of replication. But when we get into transcription, we're going to take our DNA sequence, and this is a specific gene, okay? Because again, one gene produces one protein. That one protein will express or not express the one gene, and it's all dictated by the sequence of nucleotides in that gene. So in any gene, there is a promoter region and there's a terminator region. The promoter region sounds like what it does. It promotes transcription, and terminator stops translation or transcription. And RNA polymerase is going to bind to the promoter region. It's going to nestle its way in, and it's going to unzip and unwind this molecule as it kind of binds to this promoter region. Once DNA has been opened and this RNA primase or RNA polymerase has bound to this double helix structure, it's going to work its way down this molecule until it gets to the terminator region. At that point, it's called initiation because it is actively replicating, or I shouldn't say replicating, it's not replicating DNA, but it is actively transcribing this message, okay? As it works from the promoter to the terminator, it's called elongation because the growing RNA molecule is lengthening. Then you get to termination as the RNA polymerase reaches the terminator region. And once termination has happened and this RNA polymerase has worked from the promoter down the track to the terminator, it will spit out this unprocessed RNA message, and again, this is mRNA, but it's unprocessed at this point. And it should look like this individual single-stranded ribbon that will then be processed. And so how is it processed? What do we do? DNA contains sections that are needed and sections that are not needed. Sections that are needed are called exons, and sections that are not needed are called introns. Introns will be cut out and will be removed, as you can see these sections right here are doing. Introns cut out and are kept in the nucleus. That's the way I remember it. Introns are cut out of our RNA message but are kept in the nucleus, and we know that if molecules or sections of the DNA are cut out of this RNA message and kept in the nucleus, they are not going to be expressed. And so exons, which is exactly what it sounds like, they are the portions of the DNA, which are the portions of the RNA that will be expressed. They will be spliced together. Notice that this is a shorter molecule than it was at the beginning because we've cut sections out that we are not going to be using. We added a methyl G cap, okay, a methylated guanine, and we have added a poly A tail, which is a bunch of adenines added to the back of this molecule. What is the purpose of a cap and tail? Well, it's to add integrity to the ends of these messages, okay? We want to add a cap and a tail for protection so that the RNA message that we sandwich between the cap and tail is safe from enzymes that may start trying to break these things down from the outsides in, okay? If it breaks down a little bit of this tail and it breaks down a little bit of this cap, then it's okay because we still have the important message inside. And so it buys us a little time because inevitably you're probably going to have some enzyme actions in the cytoplasm once the message has been there, okay, or once the message travels to the cytoplasm. So a little recap. RNA polymerase opens a DNA double helix, that's step 1. RNA polymerase reads each DNA base and binds to the promoter region and places in the complementary RNA bases. Now, A pairs with U in RNA, C pairs with G, G pairs with C, U pairs with A, okay, or I should say T pairs with A because we're reading DNA, so it'll contain T, but our complementary RNA bases, A pairs with U, C pairs with G. The process continues until a stop signal is reached. The process continuing, remember, is elongation. The stop signal will be reached, that is the terminator region, but that's called termination. And then you have RNA polymerase is going to leave the DNA molecule and mRNA then leaves the nucleus. But before the mRNA message leaves the nucleus, we have to process that. The way we're going to process that is we cut out introns, splice together the exons, put a poly A tail and a methyl G cap on it, and it is good to go. Good to leave the nucleus and will travel from the nucleus to the cytoplasm as an mRNA message, which will bind to a ribosome, specifically the small subunit, so that the process of translation can happen in the cytoplasm. And then what is produced by translation? Proteins, which is ultimately the goal of the DNA in the first place. I think that's where we'll leave it. Bring your questions to class. Make sure you are ready to go for our transcription review. Okay, see ya.